Hello everyone, my name is Shimekit Tadele. Welcome to my YouTube channel. For sure, you will get very interesting knowledge from this discussion. Please watch up to the end. Have a nice time. Today, I am going to discuss under general biology about biological molecules. What are biological molecules? The following are biological molecules: carbohydrates, lipids, proteins, nucleic acids, vitamins, water, and minerals. So these are the biological molecules I will discuss today. So let's discuss each of the biological molecules, starting from carbohydrates. Carbohydrates are made of atoms of carbon, hydrogen, and oxygen. Carbohydrates are important source of energy. Uh, they provide structural support for cells, help us with communication between cells, that is, cell-cell recognition as cellular markers. They use as cellular markers, carbohydrates. Carbohydrates use as cellular markers. Carbohydrates found in the form of either a sugar or many sugars, one sugar or many sugars linked together called saccharides. Carbohydrates are polyhydroxy. So hydroxy means OH. So there are a lot of OH. Poly means many. So many OH groups are there. So polyhydroxy aldehydes or ketones or substances that yield these compounds on hydrolysis. Hydrolysis means breaking down of molecules in the presence of water. For example, glucose is a carbohydrate, which is an aldehyde. It is also called aldoses. Aldoses, OS indicates it is a sugar. Which kind of sugar? Aldehyde sugar. The aldehyde sugars are reducing sugars. While fructose is a ketone, it is a ketose sugar, which is non-reducing sugar. These ketones or ketoses are non-reducing sugars. Based on the number of sugar units they contain, monosaccharides contains a single sugar molecule. A single sugar molecule. For example, hexoses, which contains 6 carbon. Hexa means 6, OS here indicates sugar. So sugar which contains 6 carbon, such as glucose, galactose, and fructose. These are the hexose sugar. They are monosaccharides. And pentose, penta means 5, which contains 5 carbon. Okay, 5 carbon. For example, ribose and deoxyribose sugars are pentose sugar, which are monosaccharides. Disaccharides contains 2 sugar molecules. Di means 2, saccharides means sugar. So disaccharides are sugars which contain 2 monosaccharides. Okay? Contains 2 sugar molecules. That is 2 monosaccharides. These 2 sugar molecules are monosaccharides. Glycosidic bond is the bond which holds the 2 monosaccharides unit together. When 2 monosaccharides come together, let's say here monosaccharide 1 bonded with monosaccharide 2, this bond is glycosidic bond. And monosaccharides are converted into disaccharides in the cell by condensation reaction. And disaccharides break down in the presence of water. By adding water, break down into 2 monosaccharide units. This is a disaccharide chemical formula. And in the presence of water, it will be converted into 2 monosaccharides. Then this process is hydrolysis. That is breaking down of molecules. This molecule will be broken down in the presence of water. That process we call hydrolysis. Hydro means water and lysis means breaking down. Therefore, breaking down of molecules in the presence of water. Oligosaccharides contain a few monosaccharides. Polysaccharides contains more than 2 sugar molecules. Examples of polysaccharides are starch, glycogen, and cellulose. The other biological molecules are lipids. Lipids are highly variable group of molecules that include fats, oils, waxes, and some steroids. Primary function of lipids is to store energy. Okay, lipids are very important source of energy. Per milligram of lipids, there will be extraction of large amount of energy. Therefore, they are a good storage of energy. Lipids are a fat if it is solid at room temperature. Fats are solid at room temperature. Fats are solid at room temperature, and oils are the lipids which are liquid at room temperature. And fat cells, also called adipocytes or lipocytes, are responsible in storing fats and lipids. To store fats and lipids, there are cells which are called fat cells. They are also called adipocytes or lipocytes. The fat cells are divided into two: white fat cells and brown fat cells. The difference is made from their ways of storing lipids. That is, white fat cells store one large drop, while brown fat cells store multiple droplets of lipids, which are spreading in the whole body of the cell. Various types of lipids occur in the human body. The various types of lipids which can found in human body are triacyl glycerol, cholesterol, polar lipids such as phospholipids, glycolipids, and sphingolipids. So phospholipids are the lipids which contain phosphate. Okay, the phosphate group. Okay, glycolipids contains uh, sugars. Okay, sugars, and the sphingolipids, the lipids which contain sphingosine. Basic structure of a lipid includes fatty acid tails. The basic structure of a lipid is the following. Here, uh, this is the head part. Okay, this is the head part which contains the phosphate group with side chain R and glycerol. The glycerol. This is the head part which is polar because of the presence of this phosphate. It is polar, and as it is polar, it is hydrophilic, which means water loving. Okay? Water loving. And the tail part, this part is the tail part. This is the tail, and this is hydrophobic. Hydrophobic, which hates water. That means it cannot react with water, as it is the non-polar portion. And each tail is a chain of carbon atoms. Okay, this is this tail. Okay? Each tail means this tail and this tail. Each tail is the chain of carbon atom. This corner indicates carbon. This is carbon, carbon, carbon. Here also carbon, carbon, carbon. So each chain is a chain of carbon atoms bonded to hydrogen and other carbon atoms by single or double bonds. Okay? Tail chains with only single bonds between the carbon atoms is called saturated. This carbon atom bonded with only one bond, this kind of bond or chain we call saturated. Saturated parties because no more hydrogen can bond to the tail. There is no free hydrogen to bond with other molecule. Therefore, it is saturated, already saturated. And tail chains that have at least one double bond between the carbon atoms is called unsaturated. At least one double bond, it is unsaturated. And uh, only single bond, okay, only single bond is saturated. Saturated. This is because it can accommodate at least one more hydrogen. Parties with more than one double bond in the tail are called polyunsaturated parties. For example, this is a double bond. This tail is unsaturated because it contains this double bond. This tail is saturated because there is no double bond. And if there are more than one double bond, let's say here additional double bond. Here is one double bond, here is an additional double bond. If there are more than one double bond, this is called polyunsaturated. Okay, poly means many, there are many double bonds. Properties of lipids. lipids are insoluble in water. Longer chains. The chains of lipids are mostly long. It becomes more hydrophobic and therefore less soluble. Okay? Less soluble because it hates water and it cannot interact with water. So less soluble. As double bonds increase, solubility increase. This is because the double bond makes the molecule unstable and therefore it will be dissolved easily. Concerning the melting points, the melting points depend on chain length and saturation. The chain length of the tail part and the saturation of the tail part. Therefore, double bonds lead acyl chain disorder, then low melting point, low melting temperatures. Importance of lipids as the main component of cell membrane as phospholipids. The lipid component in the cell membrane are phospholipids, the lipids which contain phosphate group. And other importance of lipids are insulation of heat and water, storing energy. It is an important storage form of energy, so storing energy, protection, and cell communication. These are the importance of lipids. The other biological molecules are proteins. Proteins made of small carbon compounds called amino acids. I abbreviated as AAS, amino acids. Amino acids are small compounds that are made of these atoms, carbon, nitrogen, oxygen, and hydrogen, and sometimes sulfur. All amino acids share the same general structure. All amino acids share this general structure. Here is the central carbon. It is at the center. From the outermost electron of carbon, which is 4, it can bond with 4 molecules. Okay, 4 molecules. Therefore, there are 4 compounds, 4 molecules bonded with the central carbon. The 4 molecules are, here is hydroxyl group, COOH, hydroxyl group, and the other is hydrogen, and the other is amino group, and the other is the R or side chain. The R is called the side chain. It is a variable group which makes each amino acid different. That means there are 20 amino acids. This difference of the 20 amino acids is because of the difference of this R group. That is why it is called variable group. And that means the other components, the 3 components, the carboxyl group, the hydrogen, and the amine groups are the same throughout all 20 amino acids. The 20, when you say 20, the difference is because of this R group or side chain. Peptide bonds, which are covalent bonds, join amino acids together to form proteins. So amino acids are the building blocks of proteins. Therefore, let's say amino acid 1 bonded with, let's say, amino acid 2, and this bond is what? The bond between the 2 amino acids is called what? Peptide bond. This bond is the peptide bond. A peptide forms between the amino group of one amino acid and the carboxyl group of another. That means the bond between the one amino acid is the carboxyl group, this OOH, and the NH2. The bond will be between the NH2 or amine group of one amino acid and the COOH of the other amino acid. And proteins can have up to 4 levels of structure based on the variable groups. When you say variable group, the R. So based on the variable groups contained in the different amino acids, proteins can be categorized into 4 levels of structure. Proteins primary structure, this one is protein secondary structure, proteins tertiary structure, and proteins quaternary structure. First level, second level, third level, and fourth level structures. So proteins primary structure, the number of amino acids in a chain and the order the amino acids are joined determines structure. In the primary structure, let's say amino acid 1, let me represent AA as amino acid. So amino acid 1 bond with, let's say, amino acid 2 up to amino acid n number of amino acids. Let's say primary structure is just the sequence of amino acids. Proteins are made of with a different combination and with different number of amino acids. Therefore, just the sequence of the amino acids is the primary protein structure. For example, amino acid 1 bonded with amino acid 2 bonded with up to amino acid n, for example. This just a sequence of amino acids is the primary proteins structure. And proteins secondary structure, after an amino acid chain is formed like this, after it is formed in such chain, it folds into a unique three-dimensional shape, that is secondary structure. Protein tertiary structure, a protein might contain many helices. Helices means the plural form of helix. So many helices, pleates, and folds. Many of the tertiary protein structure are globular. An example of the tertiary protein structure is hemoglobin protein. But some of the tertiary proteins form longer fibers. Some are not globular, but many are globular. The fourth level of structure or quaternary protein structure, some proteins combined with other proteins makes the proteins quaternary structure. Proteins make up about 15% of your total body mass. It is a large amount. 15% is a large amount. So 15% of the total body mass, 15% is protein. That is large amount. Proteins involved in nearly every function of your body. Nearly every function involved in nearly every function. For example, your muscles, your skin, your hair, all are proteins. Your cells contain about 10,000 different proteins. These proteins that can provide structural support, transport substances inside the cell and between cells, communicate signals into the cell and between cells, speed up chemical reactions, and control cell growth. These are the importance of proteins. The fourth biological molecules are nucleic acids. The nucleic acids store and transmit genetic information. The main function of nucleic acids is storing and transmitting genetic information from generation to generation. Nucleic acids made of smaller repeating subunits called nucleotides. The building blocks of nucleic acids are nucleotides. Nucleotides are composed of these atoms: carbon, nitrogen, oxygen, phosphorus, and hydrogen. There are three components of nucleotide, such as pentose sugar, 5-carbon sugar, phosphate, and nitrogenous bases. The three components of a nucleotide are these. That is, here is the 5-carbon sugar, that is pentose sugar, here is a phosphate, and here is the nitrogenous base. This is a double ring nitrogenous base if it is purine base, and if it is pyrimidine base, single ring. For one ring is, there are two rings, that is purine base. Either of the purine base or the pyrimidine base that we call nitrogenous bases. These are the components of a nucleotide. And there are two types of nucleic acids in living organisms. They are deoxyribonucleic acid or DNA and ribonucleic acid RNA. These are the two nucleic acids in organisms. In nucleic acids such as DNA and RNA, the sugar of one nucleotide bonds to the phosphate of another nucleotide. That means, for example, let's say this is nucleotide 1, represent it as N1, nucleotide 1, and this one is other nucleotide, say, having these components, the three components. This is, let's say, nucleotide 2, and let's say the other is here, nucleotide, the third nucleotide here, this one. Okay, this is, let's say, the third nucleotide, the nucleotide N3. So these nucleotides, one nucleotide, this nucleotide is bonding with its sugar and the phosphate of the second nucleotide. Again, here this sugar, the nucleotide, the second nucleotide sugar is bonding with the phosphate of the third nucleotide, and the chain will continue in this way. So the chain of these nucleotides will make finally a nucleic acid. It could be DNA or RNA. There are 5 different bases found in nucleotide subunits that make up DNA and RNA, such as adenine, cytosine, guanine, thymine, and uracil. These are the 5 nitrogenous bases found in the nucleic acid. The other biological molecules are vitamins. Vitamins are organic compounds that are needed in small amounts for metabolic activities. In small amounts. Many vitamins help enzymes function well. They are important in enzymes function. Vitamin D is made by cells in your skin, where vitamin made in your body is in your skin. Today's takeaway is how sunlight helps to get vitamin D. We know that when we expose to the sunlight, especially in the mornings, uh, we expect we are getting vitamin D. And our mothers expose babies in the mornings to provide vitamin D. So is that vitamin D is directly coming from the sun? I have an answer for you. When your skin is exposed to sunlight, it makes vitamin D from cholesterol, which found in your body. The sun's ultraviolet B, UVB rays hit cholesterol in the skin cells, then providing the energy for vitamin D synthesis to occur. The role of the sun is just here. It provides the energy for vitamin D synthesis. That is the sun's role. But vitamin D is not directly coming from the sun. And some B vitamins and vitamin K are produced by bacteria living in the large intestine. So bacteria are important. For example, in this sense, in this condition, uh, all bacteria are not bad. There are important bacteria. So sufficient quantities of most vitamins cannot be made by the body, by the human body. The sufficient quantities cannot be made. So a well-balanced diet should be considered, and which can provide the vitamins that are needed by the body. Some vitamins that are fat soluble can be stored in small quantities. The vitamins which are fat soluble can be stored. Other vitamins are water soluble. Water soluble vitamins cannot, okay, cannot be stored. Okay, cannot be stored in the body. Therefore, foods providing an adequate level of these vitamins should be included in a person's diet on a regular basis. The other biological molecules are water. Water molecules are formed by covalent bonds that link two hydrogen atoms to one oxygen atom. And each water molecule has the same structure. Water has the important ability to dissolve and as a media for transportation of many other substances in body, ability to dissolve. So this is the versatility of water molecule. The versatility of water as a solvent is essential to living organisms. And water molecules have an unequal distribution of charges and are called polar molecules. This means they have oppositely charged regions, that is positive charge and negative charge. The other biological molecules are minerals. These are inorganic compounds. These are inorganic compounds used by body as building material, and they are involved with metabolic functions. Example, iron is needed to make hemoglobin. Iron is what? Iron is a kind of mineral, okay, which needed to make hemoglobin. This is a protein which found in blood. It binds, the iron binds to hemoglobin in red blood cells and is delivered to body cells as blood circulates in the body. Calcium and other minerals is an important component of bones and is involved with muscle and nerve functions, and they serve as cofactors for enzymes. Calcium and others are used as cofactors for enzymes. Magnesium is an important component of the green pigment, that is chlorophyll, which is involved in photosynthesis. This is all I have for today. Thank you so much for subscribing to my YouTube channel and liking my videos. Have a nice time. Bye bye.